All right. Okay. Uh, Thanks for everyone for coming. I'm going to talk about some work I've been doing with Dr. John Honea. He's on the faculty at Emerson College. Yes, there is a little science program at Emerson College. And his background is that he worked on salmon out in the Pacific Northwest, looking at uh, dam removal and stuff like that. He was actually a postdoc with Ray Hilborn, so he's pretty knowledgeable in that field. He came to me a couple of years ago because he was interested in my alewife model that I had put together for a grant with Ben Gahagan and Mike Armstrong, which I get published, came out this past February. And he was interested in using that to look at the potential population responses of blueback herring in the Shawshene River, which is a tributary to the Merrimack River. My model wasn't really designed to do what he wanted to do, so I started from scratch with Blueback and developed this model, which I'm going to show you. Our work isn't complete yet. John had to take a hiatus because of COVID. He has to teach online, and he's got kids and everything. So this is some of the work we've done up to date. So with I think most people, when they think about dam removal, they think about trying to restore some habitat that has been closed off to some fish like the anadromous fish like the herrings and stuff, with the expectation that if we take the dam down, it opens up more habitat, and given the more habitat, we should be producing more young and have a nice positive response and population growth to what historically, hopefully what historically had occurred in that particular watershed. However, because we open up more habitat, if you will, in a given watershed, it doesn't always mean that we'll have a proportional response, because habitat quality isn't all equal within those systems, and fish exhibit preferences for habitats, and if those habitats aren't really available once they're reopened, there may not be any response at all, particularly if there's differential survival of young in different habitats, and there may not be any response given that the habitat capacity, carrying capacity, whatever, hasn't really added anything. We were, in developing this model, we were interested in how fast that a population may respond to using new habitat. And so we developed this model to look at some of the fish behavior that could be occurring once habitat is opened up. In particular, we wondered whether how fast this population could respond if, for instance, if some bluebacks or whatever, other anadromous fish, whether there is some type of homing that, given that a dam has been down for so long, and they've over decades, they've spawned in the same habitat. Even if the dam was open, would they stick around in that habitat, or would they consider moving into other portions of the river that have been opened up? We had questions on how did they explore once the dam comes down? Are they immediately responding to this new open area? Do they search around for the quality habitat that they have preference for, or is it a really slow, gradual process? Also, there's some questions we're looking at about spawning behavior and whether if in particular, like an anadromous species like blueback, we wondered if there has to be some type of minimum aggregate numbers in a spawning aggregation in order to have successful reproduction. And if, these groups, there has to be a minimum group and the population's low at the start, how rapidly could a population respond given that there's a constraint on how many they need in order to be successfully reproduce, sorry. So we had a lot of questions in mind, so I developed this model with the purpose of trying to simulate some of responses in the Shawshene River. The model is spatially explicit. Most of the relationships are empirically based. It includes all life stages and simulates dynamics over a daily time step. On the left slide, this is just the diagram of the different portions of the immature and adult populations that are modeled. There's 3 regions. They live in the ocean. They emigrate into the estuary in the spring, and then they move into the river where they spawn. The rate at which they move in is dependent on swimming speed, growth in the different areas now for the immature adults, unlike the alewife, now is all governed by bioenergetic equations, which temperature is the direct input into the growth models. And then we have separate submodels for the egg, yolk sac, and young of the year. But now, because of we wanted to look at the Shawshene River specifically, the river space is very detailed now. In the alewife model, I used R to program, but since we're dealing with some spatial, the spatial aspect, I decided to use this individual-based modeling environment known as Netlogo, which is free from Northwestern University, and you can find it on the website. In Netlogo, you have what's called a world, which you have individuals known as agents or individuals that are that will move around in this particular world. They can move, do whatever. In Netlogo, they're actually called turtles for some reason, but I think you think of them as just an individual, and they can move around in a world and interact with the environment that's within this world. So you can define patches within that world that have different characteristics. In this example, this was a case of looking at wolf predation on sheep that then the sheep are survived based on whether there's grass, which is the green, and brown, which is denuded land. So, oops. Our world is the Shawshene River, which is a tributary to the Merrimack River, and on the left is a schematic of the river connected to the Merrimack, and then the Merrimack's connected to the ocean. It's not to scale. We modeled in space, we modeled the main stem of the Shawshene River and also the primary and secondary tributaries to the main stem. There's a dam, I forget the name of it, around this area here, and below the dam there's about 88 acres of spawning habitat, or at least where bluebacks have been found. And above that dam there's about 196 acres available of different habitat qualities and stuff. Each cell in our world is 200 meters long but the widths vary depending on the location, which were based on GIS. And each cell has different water velocity and temperature based on characteristics that John developed using this book here, which was published by the USGS. This figure in the middle just shows you that one particular cell, which I coded in black, and it all has these characteristics like width, the area, velocity, temperature, and we simulate water velocity and temperature on a daily basis so that those change over time. Our agents in the model represents what I call platoons. That's from literature in the 80s, but individual-based modeling people since the 90s call them super individuals. And what a platoon just represents an agent. It's a single agent, but the agent represents more than one individual fish in this case. So in the platoon, and that's basically what a platoon literature is. So we have platoons for the mature and immature fishes, and there's 100 length-based platoons per age and sex. We keep track of sexes, ages, their length, when they enter the estuary, when they enter the river, when they leave, and when they spawn, all that, those kind of characteristics. And you can see on the right here, this is just, if you click on one of the agents, you can see all the characteristics associated with it. Once the fish spawn, the eggs within a particular cell are tracked over time, and then once they can hatch and they turn into yolk sac larvae, and then once they absorb the yolk, they become post larvae, and we keep a track of the same batch of individuals over time. This just shows an interface that you develop in Netlogo. You can have all these different graphs and stuff, which really helps because it really helps testing model and finding coding errors and things like that. So it's actually pretty neat software. So in the model, to get mature fish into the rivers, we used a lot of the Monument River data. We try to get data as close to the Shawsheen, like in the Parker and stuff like that. But when those aren't available, we move to the river that actually has data, which is mostly the Monument. So we randomly pick a day when the fish can start moving. We develop a time series of probabilities that different groups move into the river based on relationships between run start, run length, when the peak in the run occurs, and for the bluebacks, we actually did incorporate and allow males to come in earlier than females because that's been observed in many systems. For the spawning habitat, we have these individuals looking for a suitable habitat for spawning. We developed the suitability based on temperature and velocity and the values we got out of the literature. And these just show the relationships between the suitability for temperature and velocity. And this slide, if you just move from left to right, this just shows a plot of, for different days, the gradient in area, which is shows that the whitest here means the highest and black means the lowest value. You can see that the largest areas are in the main stem and the less so in the primary and secondary tributaries. Velocity, it's higher velocity in the main stem than in the tributaries. Temperature is cooler at the headwaters up at top and warmer near the mouth. And then this is just one particular day to show the ranges of the suitability that could occur. In this particular day, the highest suitability were actually in the tributaries themselves, the primary and secondary tributaries to the Shawsheen. So once the males and females are going to spawn come in, we have a set of rules for them to find the particular suitable habitat. And it's based on, in Netlogo, there's a bunch of these functions, there's a bunch of functions that allow you to search around your world depending on what you want to do. So we have it programmed so that uh, a platoon on a given day, if it's not ready to spawn, will search for the area of highest suitability within a given radius around that platoon, and that's based, that radius is based on the body length and how fast it can move in a given day. But there also, and I'll get to this later, there also has to be enough room for this platoon, the number of individuals in this platoon, to fit into that cell. And that was what I mentioned a little bit earlier. If there's, how many fish can actually fit into the cell. So we have a user specified limit they can enter. And so once they find a patch, they move there. But if they don't find a more suitable patch, they stay put. Once they move, if they can identify some spawning females, they'll spawn, and the males stay within their reaches of the river for a pre-assigned length of time, and then once that time is reached, they move back out. And for the females, they find they have the same thing. They look for the radius, around the radius, looking for males, and if when they find them, and if they're ready to spawn, they have to have the numbers before they move. There has to be enough room for them to move to the site to maintain a particular minimum ratio between males and females. So in the literature, most of the males during spawning, it's been noticed that most the ratio between males and females is skewed towards males, generally one or one female with five males. So this was our attempt to try to maintain at least a 1-to-1 ratio. It's almost impossible to maintain like 5-to-1, but there's a range of values in the literature that people have observed. So we have code just to at least maintain a minimum of 1 male to 1 female. So if there's enough room and there's enough, not enough females, they can move to that patch. Otherwise, they stay put. And if they're ready to spawn, we have them spawning only once, and then they leave the river the day after they spawn. Unfortunately, we know alewives are batch spawners, but it hasn't been really described for bluebacks, so we just had them spawn once. And for the egg and yolk sac and young of the year dynamics, we have a program so once a fish spawn and the eggs are released, they can actually drift in the system based on velocity. And there's hatching and temperature relationships, hatching success, daily mortality that control the numbers. And then when those eggs hatch, we actually have the yolk sac larvae just stay in that habitat where they're hatched. And they, once they absorb their yolk sac, they experience a carrying capacity based on the assumption that, when they first start feeding, that's the most vulnerable stage, and that the theory about mismatch hypothesis, which shows a lot of mortality because they just can't find the food or aren't used to feeding at that particular point. And there's mortality applied to them, and all the post-larva and young grow based on a bioenergetics model, and young of the year emigrate out of the system based on a probability model derived from Monument River data. This just shows, so during the model, we do a lot of calibration using different data to tweak parameters in the model to mimic what we see in the field. But there's also data sets that we don't use, and we use that as a verification. And what this slide just shows starting at the top left of the age distribution of females. And we do a pretty good job of matching that to the age distribution from the Mystic River. And then on the right is the males. And down below C and D are the length distributions for the mature fish, and we do a pretty good job at mimicking those. And at the bottom are sex ratios from the model compared to Monument, Mystic, and we're kind of in between those. And then at the bottom in F, the young of the year growth produced by the model is within the range that was observed in the Monument River by Euphrates and Oliveira. So that's pretty good. And then in the ocean, the only data we had were from our trawl survey, but also Connecticut, and our predictions of modes are pretty good, which of at least the first mode is we think is age 1. And we do a good job of predicting that, but that second mode in Connecticut Deep, I don't know what that is. That could be age 2s, but we're not matching that. In ours, we just see, and these are averages over time. We seem to mimic at least the basic large grouping of the first few length intervals. So we think it's doing a good job. So we want to look at a bunch of things, and we were wondering how population growth would occur, looking at, we have 5 different parameters. And the first one is spawning density, because that question of is, if you have a required number of fish that can fit into a particular habitat, if they have a preference, will all of those fish, if they find the highest suitable habitat, will all the fish move into that? So we looked at different levels to try and constrain how many can actually go into a particular habitat. And that has implications that if you have everything all the fish piled up into one, let's say cell, because it's the highest suitability, could that cause high mortality in young if there's like a carrying capacity issue within that cell? So we looked at 3 different levels. We also looked at ground speed, because one of the issues, as I mentioned, was how do these fish search? So if they enter the river and the dams down, and they don't explore very far because they're used to being in a particular region, or they reach a highly suitable site right away, they may not move around a lot and explore. So we looked at ground speed, different levels of ground speed, to see what would happen if you had very little movement at 0.3 and then 0.9 body lengths per second, which means that at that speed for a given day, a fish could search almost half of the river within the day. And then the third was habitat selection. So we looked at, given that those suitabilities assumes that the fish have a preference and they'll go there. In the other case, we said, well, what if there's no preference at all? What will happen? And then in the literature, there's some descriptions of how some fish respond to when dams, the response when dams come down, and particularly in some cyprinids, they're shown to essentially move right to the headwaters of that new river. Whether to spawn or not, it's not known. So we wondered in the Shawshene if when do these fish start to look for habitat, new habitat? If they start at the mouth, well, let me just back up and just say, so we wondered the start of the search, whether it occurs right when they enter at the mouth, or would they move up to the headwaters, just like some of those cyprinids and others, and then start to look for habitat. So we had a scenario for that. And then we looked at the post-larvae carrying capacity and what effects that can have. And some of our back calculation, try to develop some actual values for that, and they range from like 35 to 140 fish per meter squared for this particular river. So we looked at just 40 and 80, which 40 is low and 80 is about mid. So we did all combinations. There were 72 combinations. We simulated, we did 100 simulations per combination. And the initialization of the model, we had projected with a dam up for 30 years. We just ran the model on a base case to get the numbers of the adults and things that would be spawning, and use that as the initial values. And we kept the dam up for 10 years. We had tuned it so that there were only 4,300 fish, which we had estimated below the dam. And so we ran the model for 10 years with the dam up, and then we took the dam down, and all the different scenarios were then applied, and then the model was continued to run for another 20 years to see what the response would be. And here's just some examples of what we got. So the graph on the left is looking at the change in abundance from year 10 before the dam came down, and then compared to the abundance changes after that. The rows represent from the spawners per meter squared, but going from low in the top to high in the bottom. And then the columns represent the ground speeds for searching. And within each of those graphs, there are several lines, and you can see the legend at the bottom, where we have R1 and R2 simply means random movement and no preferences, no use of the suitability. And 1 is starting at the mouth, and 2 is starting at the headwaters, and then P1 and P2 are the preference behaviors starting at the mouth for 1 and then starting at the headwaters. On the graph on the right, the same organization, except this represents the survival ratio of the young of the year based on using the numbers of age 1 at January 1 the next year and dividing that by the eggs that were produced during that previous year. So if we start on the left, at a carrying capacity of 40, the most rapid increase was for the case scenario of R1, meaning no preference, and starting at the mouth, and slower responses for the other cases, and this is at a ground speed of 3. So what's happening is the area, they cannot take more advantage of the area at these different behaviors because the movement is so small. And if we increase the ground speed going across the columns, the differences between the first random no preference scenario, the rates of increase start to become similar as that the ground speed, the more they search and they can find areas. So there's more benefit and less crowding that occurs at the lower ground speed. And if we go over to the survival ratios, you can see what's happening. The survival of the fish at some of those other scenarios is lower for like the R2 starting at the mouth, the headwaters. You can see that's lower, so obviously it's higher mortality, lower survival, and so you don't get much of an increase. But as the ground speed increases, the mortality becomes about the same, so you get the same increase in abundance. But as you move down, going back to change of abundance, as you move and increase the number of spawners that occur within a given cell, at least for the P1 and P2s, you get a really rapid decline, and that's because these fish are clustering within a given cells, and there is a Youngnia carrying capacity. So by crowding, getting more crowded within that cell, they're producing a higher number of eggs, which are dying because of that capacity. So your mortality is a lot higher, and you can see that in the right-hand graph at the bottom for survival ratios, how that with ground speed too, if you go down the rows and then the columns, you can see how there's a much lower survival at those higher adult densities within the given cells. And so this is just showing the post-larval carrying capacity at 80. You get a much faster response. I forgot to mention that initially that there's not a response, because if they're in the first year, if the dam comes down and it's beneficial, there's a lot more habitat beneficial, you won't see any response until like 3 years, because those young who survive better aren't coming into the river until like age 3. But you can see the rates to improve in the same kind of pattern. You do get separation between the behaviors, like it doesn't really matter if you go down to the bottom row and across. You can see that the rates of increase in population changes get separated into 2 groups, and that's just the random, just searching, and then the preferences. And it's the same case here, where it's due to the individuals piling up. Under the preferences, the preferences is fewer ways to move, so they're getting piled up, so you get a higher mortality, whereas opposed to the random, they just can go anywhere and not worry about and then that mortality based on the crowding isn't really taking place. I didn't show it, but there is a difference in how the patchiness of the distribution of the individuals. So at random, with no preference, they're kind of spread out. But when there is a preference, you do get these patchiness, but at a spawning level of 0.05 individual per square meter, that clumping isn't really affecting it so much. But as you increase the numbers within a given cell, the adults anyway, that mortality is increasing. So that's as far as we got so far. John had a stop because of COVID. He's got to teach online, and he's got kids to deal with and everything. So that's as far as we got so far. So we're going to be doing some more work, but the results I showed you are probably over optimistic, because we do assume that there aren't any habitat structure changes, the same velocity occurs in the same cell, all this stuff. And we thought about trying to model the changes, but there's some quotes and people trying to do that. And one quote here by Pizzuto, 2002 in Bioscience, says it's practically impossible to try and model exactly how stream channels will change given removal of the dams. And there's actually been some surprises. Some of the publications I read looking after dam removal, there were surprises in how quality of habitat improved where they didn't think it would, and stuff like that. And then it takes a long time for fish to actually respond to changes. And in this model, we thought we model it to do an immediate response. The habitat's open. If they get to it, they can use it. Whereas that may not be true, because habitat will be evolving and changing over time. So the distribution will be affected by that, and the habitat quality will be affected like that. So we do have some big assumptions. And some of the additional work we want to do right now, there's no direct link between the preferred habitat and carrying capacity. So we're going to develop a relationship with that to see what happens. the current results that I showed you, the eggs do not drift, and then once they're laid, they just stay in that area. But we have in the model where you can have eggs drift. So we want to see what would happen if fish are preferring a particular site, the adults anyway, where those eggs go, and whether there's a benefit to that. And of course, there's no variability in the carrying capacity, which probably changes day to day and year to year. But we have that program. We didn't show that. And one of the big issues when you put the models together, you realize how little we actually know about particular species. And for bluebacks compared to the alewives, the data in the literature are pretty limited, and we need a lot more information. For bluebacks, we had to borrow parameters and relationships from alewife a lot because there aren't any available for bluebacks. So if anyone's interested in what needs to be done in terms of research, there's plenty of stuff to do. And other research could be like looking at how bluebacks are searching spawning grounds, and how do they determine where they're going to go. Do they move into one area that they think is preferred and just stay there, or they keep moving around until they're ready to spawn and then find the best habitat. Also, we just use temperature and velocity. I'm sure there's other types of habitat characteristics that they probably select. And also, densities, at least in the model, are important. So there's hardly any descriptions of densities in any rivers. So it might be a good project to try and determine what the the densities have to be or are in terms when they're spawning. So this last thing, and I'll stop. This is just the graphic showing how the model works on a spatial basis, and you can view this while you're sitting in the Netlogo thing. And I'm going to so we have all different symbols depending on the life stage and maturity and sex of individuals. So you'll see these change. And at the bottom, notice that, I don't know if you can see it, hopefully you can see it. The light blue is the ocean, and there's red and gray in there, and those represent the mature and immature males and females that stay in the ocean. You'll see all these red dots, the blue dots move into the estuary, which is the blue rectangle there. They'll stay in it for a little bit until they're ready to move in based on, I showed you the data we got from the Monument River. And then you'll see it move around. In this particular scenario, they move to the headwaters first and then do their searching. And then you'll see eggs hatch will be the pink dots, and then the yolk sac, once they hatch, they're in the yellow squares, and then once they start feeding, they become these post larvae and these little fish here. So let's see if this works. Is it going to work? Yep, there it goes. So, and then they'll move it up to the headwaters. You see them moving around in the abundance. Then the females and males will start moving out. You'll see them decrease, and then eventually, the younger the year, will start to move out based on Monument River data. And eventually, they all move out and move it to the ocean by January 1. So it's kind of cool environment if you're dealing with spatial stuff, because you can do a lot of debugging based simply on patterns that you're looking at. So that's it. That's all I got. Any questions? Thanks, Mike.
Hey, Gary, I have a question.
Mike.
So it looks like the carrying capacity for, I guess, was that the larvae or yolk sac,
Yeah, it's that
the
first
40 and
feeding
80?
mortality that we take off the top because the mismatch hypothesis stuff. Yeah.
So it looks like that was the most sensitive. The results were most sensitive to that variable.
Yeah, I mean,
Um,
that makes sense because the more that can survive in a given habitat, the more individuals you're going to get out of the population.
so do you think that well, have there been studies to sort of evaluate that at a maximum like density, or can you think of a way to devise an experiment to sort of evaluate whether that is real or whether that it's in a realistic value range? If that's the most
Yeah.
important driving variable, is there something
Well, those values we
empirical?
did use were in a range that we calculated by using the estimated number of adults in the river, some age frequency data to back calculate to get the number of eggs that potentially spawned, and then using some survival estimates in the egg and the larval stages to calculate what the post larvae had to be. And the range went from 35 to 147 fish per meter square. Um, so that's our best guess. But I don't know of anybody who's done anything like that. At least I can't recall. Maybe the real river herring people might know something.
Right. So that's a distribution of what is available to you currently.
Yeah.
But like if you're bumping up against a carrying capacity, is that are those values showing what a carrying capacity would be, or would they show you what like the distribution of densities are currently in a model system?
Mm.
Because a carrying capacity is a
Yeah.
limit; it's a
Right.
maximum.
Right. And we're saying we're trimming them. We're trimming that base. We're using this carrying capacity to say that's the maximum number of larvae that can be in that particular patch.
Right.
Um, and so to get that, then the numbers of first feeding larvae have to be truncated in order to get down to that. So that's how we're modeling that. There may be a better way of doing that, but that's the only one come up, and that's what I use in that alewife model. Um, so there has to be some type of carrying capacity, or the population would just explode and just keep going.
Right.
So that was our best guess and how to do that. Does that answer your question? I'm not really
Well,
sure.
yeah, that's why I was just, that what struck me is that that was a major driver,
Yeah.
and it, given the state of river herring currently, whether our local observations are actually relevant to carrying capacity. I'm just wondering if there's a way to sort of test that with like maybe experimental ponds or something like that,
Yeah. Yeah.
see what sort of maximum amount is possible. Because
Yeah.
it seems like that.
I think it would be system dependent too. There's a lot of, if it's based on food and prey densities and
And
stuff like
right.
that.
Yeah.
But I mean, I don't think it's ever been done in lakes. In rivers and just go and measure the density. I mean, there's like a study in North Carolina that did some plankton stuff, but to go into backwaters and stuff like that to sample if that's where the fish are going,
Yeah, you'd have to find a system where you think it would have possibility of being saturated
yeah,
by eggs to say what the carrying capacity would be.
right, right. It's difficult. I mean,
Yeah.
it's a very difficult thing. Uh, so
So what'd you think of Netlogo as far as like a learning curve and
yeah,
using it to get for your purpose?
it's bit of a challenge. There's like a dictionary that they have online and stuff that explains the functions and what they do. Um, but there's not much of a help with examples. If you wanted to do this, do this. So what you have to do, I bought several books, some of the Volcker books that do individual waste modeling using Netlogo, using their examples, but also there's a big community. And if I had questions, you could just, ask a question at the Google community thing, and that helped a lot. But a lot of stuff is just trial and error, just to get data into, our system data, like the river stuff, just to get it in. It took me weeks to try and figure it out, because they do things really different in this language. It's just not what I was used to. And the syntax is different, spaces matter, um, that kind of stuff. So you had to get used to it's a different language, really. And the way there's a function name called ask. You have to, in order to get some information off a patch, you have to ask the patch for, the information. So you have to, and then you can't ask a fish to, no, how does it? Well, anyway, there's some things you can't do between the agents and the patches unless you use a specific um, format and stuff like that. So it takes a little while to get used to it. Any other questions?
Hey Gary, it's Tracy.
Hi, Trace.
Hey, it's the model. Would you be able to put into it something along the lines of like predator effects or even competition? And I can't remember the biology well enough to remember if they compete with alewives for spawning habitat. But
well, I think there is a little overlap. Sometimes I believe bluebacks do move into lakes and stuff, and I'm not sure if alewives do spawn in rivers, but here we just have it. They only spawn in the river, so it's. I suppose we'd have to model alewives too and have different preferences and stuff like that, which to add another species wouldn't be too bad. Although the runtime on this is quite long. For one, it takes. I had for those scenarios I showed you, the 72. I had 3 instances of Netlogo running on 2 computers, and it took 2.5 weeks to run all the scenarios. So adding more information is doable, but the runtime is really going to be much longer, and yeah, I'd have to figure that out.
you need something like a bigger supercomputer or something for it.
I can't hear you. Sorry.
You would need something bigger, like a supercomputer, to add that much more complexity.
Yeah.
Yeah.
Exactly.
But the model, the guts of the software, the model that you're using can incorporate something like a predator effect.
Oh yeah. It's actually interesting. If you go to Netlogo's site, there's all these different examples of using it from different fields. So there's a bunch of biology looking at how ants track down food sources. It's really pretty cool. I recommend go and download it just to play around with that stuff, because there's some on looking at fish school, birds, how they flock, and I at one time just played around with that and made it fish schooling, and so it's actually pretty cool. And there is, although I only showed you the 2D part, they do have a 3D segment now, which I've never played around with. So that's available. So there's kind of some cool stuff associated with the environment. So, and I see David had a question. Why does the mode of the ocean, Connecticut, not seem to match the second peak? I have no idea. I don't know if that's a real peak.
Yeah, can you hear me?
Yeah, I can hear you.
Yeah. So in one of your one of the plots that you had up there, right? It had the model overlaying the bins there, the population bins, and it didn't look like the model fit that second mode at all. Yeah.
Yeah, I have no idea what that is. We don't usually see that too often in our survey. So again, I try to borrow data wherever I could get it, and I'm sure it won't exactly match. It could have been a mixture of alewives and bluebacks and that. I don't know.
Thanks.
Yeah. Anyone else?
Gary, this is Sam. I've got a quick question. Can you hear me?
Yeah.
Okay, I was just wondering how you arrived at your final suitability for each cell. You have the variables area, velocity, and temperature. And I was just wondering, are those equally weighted, or is the suitability some function of those three?
We didn't do area, we just a cell. So you have this the relationships between the temperature and velocity. So on a daily basis, they can change in a cell. So once they do, we go back and use those relationships to get the suitability for each of those cells. So it changed individually, if that makes sense.
Okay. Well, maybe I'll just catch up with you. I just didn't quite follow, but thank you.
Okay. Anyone else? All right, thanks. So next week, who's up? Oh, Scott Elzey is going to be talking about or giving an overview of the age and growth program. So come back next week. Thanks everyone for attending. Hope everyone's well.